Hello everyone, my name is Aslı. I'm currently a master's student in Neuroscience Department at Bilkent University, Turkey. In this presentation, I'll be talking about my recent research on motion perception. Medial temporal lobe, where motion perception is processed, relies on surround modulation mechanisms to perceive and discriminate motion. However, low-level features of a stimulus such as size and contrast may change our ability to perceive motion. As the size of stimulus increases, if the contrast is low, receptive fields around facilitate the activation of center neurons and our ability to discriminate motion gets better. Whereas if the contrast is high, surround neurons suppresses the activation of center and therefore our ability gets worse. In previous literature, researchers studied this phenomenon with grating stimuli, which were small and large disks of gratings. They studied various contrast levels and sizes and found the same effect of facilitation in low contrast and suppression effect in high contrast. What was missing in the literature, to our knowledge, was the study of surround structures as an annulus aperture, meaning that research has only focused on the receptive field as a whole, like 5 to 12 degree disk gratings, and sometimes focus on the center via small disks such as 1 or 2 degree disks. Therefore, in this study, we wanted to focus on covering the surround portion of the receptive field via annulus apertures and see whether it would enhance or hinder motion perception. To do that, we decided to subtract centers from whole RF structures to get an annulus aperture, which represents surround portion of an RF. In terms of size, we take 1 to 2 degrees for this inner diameter and 5 and 12 degrees for this outer diameter portion. In each trial, participants start with a fixation point with a mid-gray background. They were asked to report motion direction of the stimulus and press left or right key on a keyboard. For each stimulus type, there were 80 trials. As for the results, there was an interaction effect between the size and contrast, meaning that for different levels of size and contrast, threshold levels differed systematically. As you can see in this graph, in low contrast condition, threshold levels differed systematically. There was a drop in threshold levels going from 1 to 2 degree inner diameters, and for both 5 degree and 12 degree outer diameters. This means that in 2 degree inner diameters, surround facilitates the center more. Additionally, in 12 degree stimulus type, there was an additional suppression effect. This is a suppression effect we see in low contrast stimuli as the size increases to these levels. Finally, we can see that there were no changes in high contrast in any of the size conditions. So to discuss these results, a facilitation effect appears in the conditions where it goes from 1 to 2 degrees. After this drop, they're nearly equal. As you can see here, after they drop, they're in the same level. There are no different changes, no differences in high contrast condition, and we see this additional suppression effect as the previous studies have shown. So there might be some limitations to our study. First one might be due to the participant number being low, which was 8 participants. Another one is about attention. According to one study, if the participant narrows down their visual attentional field, suppression effect of surround might also diminish. Although participants were fixated on a point, they had to discriminate the motion direction of the stimulus and therefore widening their attentional field. So this might not apply, but still it might be something to consider. For future directions, to better understand underlying mechanisms and brain activation, task-based fMRI studies could be carried out with an addition of attention as a condition. Finally, takes to ISBCS community for giving me this opportunity. I also want to thank my advisor, Hüseyin Boyacı, and my friends at my lab for participating in my study. Thank you for hearing me out, and I wish you are all healthy and safe. Goodbye.